Today we are going to talk about insulin receptor and signal transduction. This lecture is basically a summary of previous 4 lectures on insulin receptors and signal transduction. What I want to say that already we have recorded 4 lectures where we explain in detail what is the structure of insulin receptor and how it gives intracellular signaling. But here in today's lecture, we are going to have a sum and summary of previous 4 lectures. So this lecture will have a little faster pace. Let's start with it with very briefly. When you take a nutrient-rich meal, right? Let's suppose you have taken a meal which is very rich in carbohydrates and amino acids, and nutrients are absorbing from your GIT. These nutrients, as you know, will stimulate, yes, beta cell of pancreas, and beta cells of the pancreas will release insulin, right? Now you know insulin, what is the main purpose of insulin? Insulin is basically a very powerful anabolic hormone. It's a very powerful anabolic hormone. One of its major function is that all the nutrients which are coming, it has to pump those nutrients into body tissues, especially in the liver, skeletal muscles, and lipocytes, right? So glucose, amino acid uptakes should go in the target cell, and those nutrients should be utilized in the cells and stored in the cells, right? So what insulin is going to do? What is the basic function of insulin? Basic function and purpose of insulin is that glucose, amino acids, and other nutrients which are absorbed, especially glucose, right? It should help the cell, that cells should take up the glucose and amino acids. It means insulin changes the membrane permeability, membrane characteristics. Then once the nutrients are in the cell, then insulin also modifies the enzymatic functions. It changes the metabolism of the cell in such a way that nutrients should be more utilized and more stored. And in the end, insulin also acts on the nucleus. And when insulin acts on the nucleus, right, it changes the expression of genes, because insulin plays a very active role in cell proliferation, growth, differentiation, and survival. So I will repeat it, that insulin on the target cells has to act at 3 levels: on the membranes for uptake of nutrients, altering the metabolic machinery, and altering the genetic machinery, right? But there is an issue. The issue is this: insulin is basically 51 amino acid molecule, and this molecule, which consists of 2 peptide chains held by 2 disulfide bond, this peptide is relatively a larger molecule, and it is highly polar molecule, highly charged molecule. So to produce its actions, it cannot enter into target tissues. To be more accurately, to produce its action, as it is larger molecule and highly charged molecule, it cannot enter into target cells, because if insulin has to enter into target cell, it has to be a smaller molecule and it has to be lipophilic, not charged molecule. So it means that insulin will remain outside the target cell membranes, but still produce action on the membranes, action on the cytosol metabolism, and action on the nucleus. Is that right? Mainly insulin, as you must be knowing, act on which tissues? Yes, primarily it acts on the liver, yes, striated muscles, with skeletal muscles and even myocardium, and also lipocytes. These are the main targets of insulin, right? Now, all these tissues have insulin receptors. Let's suppose we have a hypothetical cell here, and this cell is having an insulin receptor, and we will find that when insulin binds, how this receptor is activated and gives intercellular signals. Basically, insulin receptor is a tetrameric molecule. It consists of 4 peptides. Insulin receptor, it has 4 parts. One part is, yes, this is extracellular part, These are two parts, right? Two peptides, and these two peptides are attached with two more peptides which are, yes, right. Now this is insulin receptor structure. It has these two peptides which are called alpha subunits, and these two peptides, these are called beta subunits. These are called beta subunits. We can imagine that insulin receptor is something like a jungle queen, right? Jungle queen with two arms. These are two arms, and these are two legs. Rest of the things I don't want to show, right? So the insulin receptor is just like a jungle queen, but this queen legs are inserted into membrane, arms are outside. So we can remember for this jungle queen molecule, alpha for arms, these are alpha units, and what are these units? Beta units. Beta for beautiful legs. Beta for beautiful legs. So this jungle queen, this molecule is just like a jungle queen having two arms and two beautiful legs. Arms are totally extracellular. These two alpha units are totally extracellular, and legs are transmembrane. A part of the legs, part of the beta component is extracellular, part of it is passing through the membrane, and part of it is intracellular. So we can say beta subunits are transmembrane. Now, each alpha unit is held with beta unit through, what are these bonds? Disulfide bond. And even two alpha units are also held with each other through disulfide bond. So we can say that insulin receptor is inserted in the membrane having two alpha units, two beta units. Alpha units are extracellular, beta units are transmembrane, having one component extracellular, other component through the membrane, and another component intracellular. Is that right? Now, when you have taken the nutrient-rich meal, especially carbohydrate-rich meal, insulin is released, and these insulin molecules are flooding in the blood and reaching to the target tissues, right? And eventually, insulin molecules will bind with alpha units of insulin receptors. Where will they bind? Alpha units of insulin receptors. Suppose this is our insulin, happily coming in the hands of jungle queen, right? Now, once the insulin bind with, what are these alpha units, right? Alpha units undergo a change. Let's suppose hands were like this. When they will hold insulin, the hands will become active. So when insulin bind with alpha subunits become active. They undergo transformational change. Alpha units undergo transformational change. And when they undergo transformational change, they induce a transformational change in the beta subunits. So what has happened? Insulin bind with the alpha units. Alpha units underwent transformational change, and that transformational change induced transformational change in beta units. When beta units are activated, because once insulin is in the hands, it means there's a lot of nutrients. It means there are a lot of nutrients. And jungle queen is sad or happy? Happy. Having the insulin. Insulin is the crown. She's having the crown in her hands, and hands will lead to stimulation to the legs, right? And when these legs are stimulated, right? Yes, now she is happy. Before, when there was no insulin, right, these legs were very sad. Legs can be sad also. I never knew, right? So, when beta subunits are stimulated, actually legs become hot legs. Don't ask me to the detail of hot legs. Just imagine that the legs become hot legs. These are beautiful legs, these are hot legs, and once these are activated, there is a special type of, yes, there is a very special type of enzyme inside the beta units. Now what I mean by these enzymes, let me explain it. Within this beta peptide, there is intrinsic, what is there? Intrinsic enzyme. And this enzyme is able to bind phosphates on tyrosine residues. This enzyme, which is the integral part of, intrinsic part of, internal part of beta subunits, this enzyme has a special function. What is the function of this enzyme? This enzyme can bind phosphates with tyrosine amino acids. So basically, this enzyme is tyrosine kinase, or we can say that beta subunit intracellular part of beta subunits of insulin receptors have intrinsic tyrosine kinase activity, and once insulin bind with alpha units, we stimulate the beta units, and that will result into activation of intrinsic tyrosine kinases. Now, once these intrinsic tyrosine kinases are activated, what these enzymes will do? These enzymes will, let's suppose, this is my one hand, these are the legs. There were hands here, insulin here, and now these are the beta subunits. When beta subunits are activated, tyrosine kinases within them is activated. Now, tyrosine kinase of one peptide, beta unit, will attack the other and look for where is tyrosine residues. This beta unit is made of many amino acids. So, one beta unit will come close to the other beta unit. One beta unit will come close to the other beta unit and start looking where is the tyrosine amino acids. And once it finds it, it will plaster their phosphate. I mean, it will take the ATP, convert that into ADP, and phosphate will be bound with the tyrosine residues of other beta subunit, and we say there is tyrosine phosphorylation. So, we can say if this is the tyrosine residue here, this is another tyrosine residue here, this is another tyrosine residue here. I am showing 3 tyrosine residues here and 3 tyrosine residues here. So, what is happening that basically these tyrosine kinase enzyme from one beta unit will attack the tyrosines of other and lead to their phosphorylation, right? And then the second beta subunit will attack the first one and what do? Phosphorylation of tyrosine residues. Now we can see it that once insulin bind with alpha units, stimulated alpha units stimulate the beta unit, within the beta unit tyrosine kinases are activated, and then these tyrosine kinases, which are part of the receptor, right, lead to phosphorylation of their own tyrosine residues. So this should be called autophosphorylation. So when insulin binds over here, we say when insulin bind with extracellular alpha subunits, intracellular part of the beta subunits undergo autophosphorylation. And because one beta is doing phosphorylation of the other beta, so we say this autophosphorylation is also cross phosphorylation. And some books mention, yes, some books mention transphosphorylation, right? So once it becomes phosphorylated, we say these hot legs become super hot. These hot legs become super hot, right? Now these super hot legs of this jungle queen, they will love to, yes, what hot legs want? Phosphates are here. So not only these are hot, hot means they are stimulated, right? But they are also phosphorylated. They are hot and sticky. What they want? What they want to do? Dance. So now these legs, beta subunits, internal, they want to dance. They want to give signals, right? And for dancing, they need a dancing floor. They need a dancing floor. In the cell, within the cell cytosol, there is a protein which acts as a dancing floor. So what happens that there is a protein here, right? And this protein, if there is no insulin, the receptor is no more active and there is no phosphorylation here. There is no autophosphorylation of receptor, then this protein cannot bind over there. But as soon as insulin comes, and internal part of the receptor beta subunits undergo phosphorylation, these become hot and sticky, and then this cytosolic protein can float there and stick on it, right? And this cytosolic protein, when it will float and stick over with the phosphorylated what? Tyrosine residues, right? This is our dancing floor. These legs are dancing over this protein. And what should be the name of this protein? It is basically a substrate. This protein is a substrate for the active phosphorylated insulin receptor. This is a substrate for the active phosphorylated insulin receptor. So its name is, yes, its name is insulin receptor substrate. There are many types of insulin receptor substrates depending upon the tissues, right? But I'm just mentioning that as soon as beta components are phosphorylated, right? Phosphorylated component will attract, recruit insulin receptor substrates. These substrates will bind with these phosphorylated tyrosine residues of beta subunit. Once this IRS binds over here, you know that these hot and sticky legs are very naughty legs. These are very naughty legs. Now these legs will start action on the IRS. This enzyme, what was the name of this enzyme? Tyrosine kinase. First this enzyme autophosphorylated the tyrosine residues on its own beta units and then attracted what? IRS, and then it will start phosphorylating tyrosine residues of the IRS. It means IRS also have tyrosine residues. IRS also has tyrosine residues, and what is this enzyme? Tyrosine kinase. So what will happen? This tyrosine kinase, which initially attacked each other, then it will go and attack IRS and phosphorylate the tyrosine residues on IRS, right? There are 20 to 22 tyrosine residues on IRS will be phosphorylated, right? And when IRS is phosphorylated, it is ready to give signals, right? Jungle queen, hot legs, phosphorylated beta units leading to phosphorylation of IRS, dance floor is prepared, right? And who should come there? Jungle queen is so happy. Remember, when this protein undergoes phosphorylation, when this protein IRS undergoes phosphorylation, this protein undergoes alteration. It changes its configuration and it expresses to very special areas, to very special domains. It means once IRS is phosphorylated by the activated insulin receptor, IRS undergoes a change, conformational change after the phosphorylation, and it expresses to two domains, two hands, right? This dance floor has made two extensions, right? It is calling someone. Who should come there? Who should come there? Yes? Who should come there? When jungle queen legs so hot, so sticky, phosphorylated, even dance floor is phosphorylated, who should come there? Tarzan should come there. It's very logical. So a Tarzan protein will come here, and this protein, this Tarzan protein will bind over here, right? Very happy. Right? And another protein will come on this side. That I will discuss later. First I'm going to talk about this pathway, because from here many signals will go. Through the IRS, many signals will go. But I will talk mainly two pathways. One pathway which goes and changes in the membranes so that glucose and amino acid, phosphates and potassium should be uptaken. And also this goes to changes in the metabolism of the cell. And other pathway will send the signals to the nucleus. And there are some other, but we will focus on these two. Now, this is Tarzan 1. This Tarzan 1 protein come over here. It will only bind here when IRS is phosphorylated, and IRS will be only phosphorylated when beta cells are phosphorylated, and they will be only phosphorylated if alpha units have insulin. So insulin there, whole signaling pathway, first I will discuss this. This Tarzan is here, right? And what this Tarzan will do? Actually, it is going to act on special lipids which are present in the membrane. It's going to act on special lipids which are present in the membrane. Now, what are those special lipids? Let me show them here. These lipids have fatty acids, long chain fatty acids with glycerol. These are fatty acids, and these are glycerol backbone with one phosphate here and one inositol molecule here. What is this? Inositol molecule here. This lipid, which is one of the component of the lipids present in the internal lamina of the cell membrane, this is a very special lipid, right? Tarzan is going to interact with this lipid, right? Now, this, what is the name of this lipid? Basically, this lipid has two long chain fatty acid. What is this? Glycerol backbone. There is a phosphate here, which is component of this lipid, and this is inositol, and this inositol has already two phosphates attached with it at carbon number 4 and carbon number 5, right? Now, this unit is called, yes, what is the name of this unit? This unit is called phosphatidyl inositol diphosphate, because this lipid unit is phosphatidic acid. What is it? Phosphatidic acid. And then what is this? Inositol having phosphate at position number 4 and 5, having two phosphates. So this molecule, red molecule, it is called phosphatidyl inositol diphosphate. And to be more specific, phosphatidyl inositol 3,4 bisphosphate. 3,4 bisphosphate is 4,5. Oh, thank you. It is 4,5. It means no one is sleeping here. Thanks God. So what is this? Phosphatidyl inositol 4,5 bisphosphate. Is it clear? Now, what this Tarzan will do? Tarzan is an enzyme which will act on this lipid, and Tarzan will add their, yes, Tarzan will add their one more phosphate, right? So this lipid will undergo a change. This lipid will undergo a change, and that change will result into modified lipid having the same long chain fatty acid with glycerol backbone with phosphate inositol. This is inositol. Now, this inositol was previously having 4 and 5 phosphates. Is that right? Now, this Tarzan enzyme will add one more phosphate at position number 3. So it means this is, what is this enzyme? This is an, what kind of enzyme is this? This is a kinase. Why I say this is kinase? Because this enzyme has done the kinase activity on the PIP2. This enzyme has done kinase activity on PIP2. It means it has added a phosphate to PIP2, and now it has how many phosphate? 3 phosphate. So now this enzyme, this lipid will be called PIP3, phosphatidyl inositol triphosphate. To be more specific, actually it has added the phosphate at carbon number 3. So we will call it that this product is phosphatidyl inositol 3,4,5 trisphosphate. Is that right? Tri or trisphosphate. Is it clear? So what is the function of this enzyme? This enzyme converts the phosphatidyl inositol 3,4,5 bisphosphate into phosphatidyl inositol 3,4,5 trisphosphate. Is that right? So what should be the name of this enzyme? The name of this enzyme, generally, this is an enzyme which is doing kinase activity on a lipid. So generally speaking, we can just say that this is lipid kinase. But to be more specific, what is the name of this enzyme? This enzyme is phosphatidyl inositol 3 kinase. Why it is given so name? Because this enzyme acts on the phosphatidyl inositol product at carbon number 3 and do kinase activity. 4,5 was already there. So because function of this enzyme is that it will catch phosphatidyl inositol molecule and do kinase activity at carbon number 3. This was carbon number 3. So this is called Pi3k or phosphatidyl inositol 3 kinase. Now here I would love to mention one more thing. When IRS was phosphorylated, it was activated, it expressed special domain. Now these special domain which were expressed by the activated phosphorylated IRS, this will bind only special domain of next protein. Is that right? If these components were not active, can Pi3k bind over here? No. But even if this is active and this component of the Pi3k is not there, can still bind? No. So this component of Pi3k which is interacting with phosphorylated what? IRS, this component, yes, this component is called SH2 domain. What is it called? SH2 domain of Pi3k which binds with the phosphorylated IRS. Is that right? Now SH2 does not stand for sulfhydryl here. Here it stands for Sark homology 2 domain. Sark homology 2 domain. SRC, Sark homology 2 domain. So it means when IRS is phosphorylated with the phosphotyrosine component of IRS, the proteins which bind, those proteins must have Sark homology 2 domain. Is that clear? Let's recap. What is this? Insulin interacting with alpha subunits of insulin receptor. Alpha subunits change and produce change into beta subunit. Beta subunit activated and what is this? Tyrosine kinase enzyme lead to autophosphorylation of tyrosine residues of beta subunits of insulin receptor. And this autophosphorylated receptor can recruit and bind IRS. And once IRS bind over there, then tyrosine kinase will now attack IRS and lead to tyrosine phosphorylations of the IRS. Once IRS is phosphorylated, now IRS is ready to bind other proteins. Now IRS is ready to bind other proteins. Which proteins? Those proteins which have SH2 domains. Those proteins which have sulfhydryl. Very right. Not sulfhydryl. This is Sark homology 2 domains. And once these proteins bind, one of the protein which bind here is enzyme lipid kinase phosphatidyl inositol 3 kinase, which will act on phosphatidyl inositol 4,5 bisphosphate and do kinase activity and convert that into phosphatidyl inositol 3, 4, 5 tris phosphate, right? Now, any problem up to this? So what has happened? Arrival of insulin, in a very short statement, arrival of insulin and interaction with the receptor and receptor interaction with IRS and IRS activation of Pi3k resulted into conversion of phosphatidyl inositol bisphosphate into phosphatidyl inositol 3-phosphate. From here, a very important component comes. These three phosphates are dangling like coconuts. This is a jungle, you know. These are dangling like, hanging like coconuts. When these coconuts are hanging, who will come there? Monkeys. Yes, monkey should come after the coconuts, right? And some naughty boys too, right? So what will happen? There were two types of enzyme will come. One, naughty boy, right? And other is monkey. Monkey will bind also. Right? Now this is our monkey. Now what is happening now? That arrival of insulin and stimulation of its receptor through a molecular machinery has generated lot of PIP3 in the cell membranes of the target cells, right? And once PIP3 is produced in the target cell, these target cell may be muscle cells, or these target cell may be adipocytes, or even now what will happen that PIP3 will attract two cytosolic proteins. One is naughty boy. Why I call it naughty boy? Because it is really naughty. As soon as the monkey come here, both of them are after the same coconuts, PIP3. Monkey boy will stick the banana into monkey. The naughty boy will stick the banana into monkey. So what happens that this will, once both of them are added together, this naughty boy will act on the monkey and monkey will have banana. Where I should add the banana? Okay, let's suppose this is the banana. This is unusual type of banana. Okay. Right? Once this monkey gets the banana, it's sad or happy? Happy, you know, love the banana. Monkeys love the bananas. You offer them dollars, they don't care. Right? They love bananas. This monkey will, as soon as it is banana-fied, right, it gets the banana, this monkey will happily jump around and get lost into jungle. Leave this membrane area. Then more monkeys will come, more monkey molecule will come, naughty boy will keep on adding the bananas, and banana loaded monkeys will run away for their ultimate action in the cell. Right? Now what is this monkey? Because monkey is the real champion of the jungle. Because once it is activated by receiving the banana, it will leave this membrane component. Because whole this insulin receptor and this molecular machinery was along the membrane, cell membrane. On the inner lamina of the cellular membrane, this is the first molecule which will get active and run away, where? To the cytosol for multiple actions. You know, monkeys are very multifunctional characteristics. Now, what is this monkey? This monkey is basically a very important molecule which is called AKT protein. Monkey in action AKT, right? And another name for this is protein kinase B. Another name for this monkey is protein kinase B. So basically this is protein kinase B AKT molecule. You can write it like this: protein kinase B / AKT, because in different literature they mention different, but they are one and the same thing, right? Now protein kinase B or AKT molecule, what is this banana? Again phosphorylation, Once this molecule gets phosphorylated, it becomes active and goes for its action. But what is this? This is a kinase, because it is adding the phosphate over there. Any enzyme which is adding phosphate to other side is kinase. Now, what is the name of this kinase? The name of this kinase is PIP3 dependent kinase, because this kinase is only activated, this naughty boy is only activated when membrane is having PIP3, right? This naughty boy is basically PDK1 molecule. What is this? PDK1 molecule. Why we call it PDK1 molecule? Because this is a enzyme which is phosphatidyl inositol 3 dependent kinase, right? And it does kinase activity on the AKT and lead to phosphorylation of AKT/P.K.B. Is that right? And more AKT molecules come over here, get phosphorylated and active, and they go into cytosol for their ultimate actions. Any question up to this? So what we learned, insulin came over here, and through this all molecular machinery, it's generating in the cell lot of protein kinase B/AKT molecules which are phosphorylated. Now what this monkey is going to do? Let's come over this monkey here, right? This is a happy monkey or sad monkey? This is very happy monkey. And phosphorylated, of course, and this banana is phosphorylation. Now it will start its ultimate action. One action is that it will act on the intracellular vesicles which are loaded with special glucose transporters, and these glucose transporters are called Glut4, glucose transporters type 4, right? And they will act on this transporter, they will act on these vesicles through multiple molecular pathways. Actually, Akt protein will phosphorylate multiple molecular motors, and these molecular motor will drag this vesicle to the membrane. They will drag this vesicle to the membrane, and this vesicle is loaded with Glut4 molecules or glucose transporters number 4. And once AKT level in the cell goes up, all these vesicles loaded with this are pushed to the membrane, and when they are pushed to the membrane, what will happen? That they will move towards the membrane and fuse with the membrane. This vesicle will move towards the membrane and fuse with the membrane, and now these Glut4 molecules will become part of the membrane, and now membrane become permeable to glucose, and then what will start jumping in? Glucose. Glucose is coming in. So this is first action which I want to highlight, that AKT molecules, right, once they are phosphorylated, they have many functions. First important function which I'm mentioning is they will act on the intracellular vesicles loaded with glucose transporters for Glut4, and these vesicles will travel to the cell membrane and fuse there, and their Glut4 molecules will be inserted in the membrane, and membrane of this target cell will become permeable to glucose. This action is especially seen in skeletal muscles and lipocytes, adipocytes. So what happens when you have taken food, right, especially rich in carbohydrate and amino acids, insulin levels in the blood go up, insulin act on its receptors, right, on liver, muscles and fat, especially in the muscles and the adipocytes, right. Glut4 molecules will come on the surface and glucose will start moving inside. So this is one function which insulin has achieved, that nutrients which were extracellular, now they are able to move in, right, and glucose uptake is enhanced. Any question up to this? And not only glucose uptake is enhanced, I will mention not only glucose is coming, even the pathways, transport mechanism for amino acid uptake are also enhanced. Even pathways for potassium uptake and phosphate uptake are also enhanced. So we can say that once insulin levels in the blood go up and they stimulate the receptor and intracellular machinery through this pathway. This pathway is called Pi3k P.K.B pathway, or this is Pi3k P.K.B / Akt pathway. Is that right? This pathway, within, once insulin bind here, within seconds to minutes, membranes change their characteristics, and lot of glucose, amino acids, potassium and phosphate that start jumping into cells. Any question up to this? After that, what will happen? Let's suppose what happens with this glucose which is coming in. This glucose which has come in. Actually, especially in liver cells, it will activate an enzyme. Akt will now act as another function. It will activate an enzyme which is called glucokinase. And not only activate this enzyme, even expression of this enzyme will enhance in the cell. And glucokinase will lead to phosphorylation of glucose. So, what is happening? The glucose which is coming in, that is getting more and more phosphorylated. And when this glucose is getting phosphorylated, concentration of free glucose in the cell is decreasing and more glucose will come in. So it means that this Akt protein not only helps the insulin sensitive cells to take up more glucose, even it helps as glucose to convert into glucose 6-phosphate. And once glucose into glucose 6-phosphate, now it can go along the which pathway? Carbohydrate metabolic pathways, right? This glucose will be now utilized for glycolysis. So cells will use this glucose for the energy. This glucose will be used to enhance synthesis of glycogen. Let me tell you how synthesis of glycogen increases. How the synthesis of glycogen increases. Another action of our naughty monkey. Now this monkey has done one action over here. What was this? It has increased the permeability of cell to the glucose. Number 2, it is the glucose which is coming in, it is utilized for what? Glycolysis. Then it will do one more thing. What is that? This Akt will phosphorylate an enzyme which is called glucose GSK3, glycogen synthase kinase 3. Now let me tell you, stop it here. Normally what happens? There is glucose here. Glucose is acted upon by a very important enzyme which is called glycogen synthase. It's a very simple concept. Their glucose molecules are acted upon by many enzymes. I'm just showing one important enzyme. What is this? Glycogen synthase. Glycogen synthase. This glycogen synthase makes the polymers of glucose. It is one of the enzymes, and glucose converted into glycogen. Now, when you have taken carbohydrate rich meal and glucose level in the blood is high, cells should take up less glucose or more glucose? More glucose. That is what insulin is doing. Should it use glucose for energy? Yes. Again, glycolysis is fast. Plus extra glucose should be stored as glycogen. It means glycogen synthase should be working less or more? It should be working more. Now, when blood glucose level is high and insulin level is high, intracellular machinery should activate the glycogen synthase. Now, glycogen synthase is very funny. If it is phosphorylated, if glycogen synthase is phosphorylated, if glycogen synthase is, let's suppose here, phosphorylated, it is inactive. What is it? Inactive. If it is not phosphorylated, then it is active. Again, listen. If I am the glycogen synthase, if I am phosphorylated, I am inactive. But if I get dephosphorylated, right, or I am not phosphorylated, I will start converting glucose into glycogen. Is that right? Now listen here. What happened? GSK3, come back to this. This monkey Akt or protein kinase B will phosphorylate GSK3. Right? Now GSK3, what is the function of this? This is glycogen synthase. What is this? Glycogen synthase. And this is glycogen synthase kinase. So it does the kinase activity over here. So if GSK3 is active, it will phosphorylate glycogen synthase. If GSK3 is active, it will phosphorylate glycogen synthase. And if glycogen synthase is phosphorylated, will it be active? No, it will not be active, right? But what we want, glucose is coming, glucose should convert into glycogen. So what really happens? This monkey lead to phosphorylation of GSK3. When this Akt protein, phosphorylated Akt of protein kinase B, phosphorylated, lead to phosphorylation of GSK3, GSK3 becomes phosphorylated. And when GSK3 is phosphorylated, it becomes inactive, inhibited, it becomes disabled. So Akt basically disables the GSK3. When GSK3 is disabled, can it phosphorylate GS? No. So it is unable to phosphorylate glycogen synthase. And now glycogen synthase, which is not phosphorylated, it will start converting glucose into glycogen. It means now what will happen in the cell? Glycogenolysis. No. Glycogenesis. What will happen in the cell? Glycogenesis. Now let's see what functions should happen. Let's come over here, metabolic functions. As I said, these molecules are basically, insulin is anabolic hormone. Let's suppose talk about carbohydrate metabolism, protein metabolism, very briefly I am discussing, and lipid metabolism. Details we will discuss in other lectures. Now, up to now, insulin action has produced, number one, increase glucose uptake. Number two, it has led to the increase glycolysis. Plus, due to phosphorylation of GSK3, disabled or inhibited GSK3 cannot phosphorylate glycogen synthase. Glycogen synthase activity goes up, and that will lead to what? Glycogenesis. So we will write here, there is increased glycogenesis. Is that right? Now, when nutrients are more, we should make glycogen more, right? But do you think that we should need more breakdown of glycogen at this moment? No. When a lot of glucose is coming, we don't want to break down the previously made glycogen. So glycogenolysis should be inhibited. So we can say glucose uptake is more, glycolysis is more, glycogenesis is more. But other enzymes which are related with catabolism, they will be inhibited, because anabolic hormone should stimulate the enzyme for anabolism and inhibit the enzyme for catabolism. So anabolic hormones are activated like glycogen synthase, and catabolic enzymes, right? In the glucose, what are the catabolic enzymes? Enzyme for glycogenolysis, which break down the glycogen, and enzyme which are for gluconeogenesis, which make the glucose. These enzymes, which are part of the gluconeogenic pathway, they synthesize the glucose from where? From carbon backbones of amino acids and even lipids. So we can say in gluconeogenesis, cells are making glucose from non-carbohydrate sources. Now, these enzymes should be inhibited. So these enzyme system is inhibited by reduced synthesis of enzyme and special phosphorylations of enzyme. Here I want to mention one thing. Let's suppose this is one enzyme, right? If it is phosphorylated here, this mouth will open more and it will become active. But there is another enzyme, let's suppose, and this is phosphorylated inside, this will become inhibited. So phosphorylation can activate some proteins and inhibit other protein, depending upon that how phosphorylation alter the active domain of that protein. So this Akt, right, it is acting on multiple enzymes in such a way that glycogen synthesis is increased and glycogenolysis is decreased and gluconeogenesis is decreased. Now we come to protein. What do you think if it is anabolic hormone? Protein synthesis should go up. So protein synthesis should go up and proteolysis should go down. In the same way, lipid synthesis should go up and lipid breakdown should go down. Lipolysis should go down. Now, how these things are achieved? For these I told you, the GLUT4 went into membrane, glucose came, glucokinase in the liver was activated and glucose was trapped, and especially muscles and the liver, glycogen GSK3 was phosphorylated, inhibited, so it was unable to phosphorylate glycogen synthesis. So unphosphorylated glycogen synthesis was starting glycogenesis. Is that right? And then I will not go into detail, but some other phosphorylations will lead to inhibition of enzyme related with glycogenolysis and gluconeogenesis. Now, protein and lipid. Actually, how protein synthesis and lipid synthesis is enhanced? Because when insulin is there, it means nutrients are more. Not only we have to synthesize, because anabolic action, not only we have to synthesize the glycogen, we have to also synthesize more protein and more lipids. For this purpose, again this monkey will, through multiple pathways, will phosphorylate a protein, very funny name, mTOR. mTOR is mammalian target of rapamycin. I don't know why they put this name. Maybe the rapamycin act on a special protein and a mammalian protein, and they say, okay, this protein is just called mammalian target of rapamycin. Later on they discovered this protein basically enhances the lipid synthesis and also enhances the protein synthesis and also activates the enzyme related with lipid synthesis. So it means mTOR pathway will increase protein synthesis in the cell and lipid synthesis in the cell. So how much we have learned? We have learned that insulin bind with alpha receptors, alpha subunits of the insulin receptor which activate the beta subunit. Activated beta subunits have activated intrinsic tyrosine kinase, which first lead to auto phosphorylation of beta subunits, tyrosine components. Then auto phosphorylated beta subunit bind IRS, then IRS tyrosine units undergo phosphorylation. Then this phosphorylated will be able to attract Pi3k kinase, right, or phosphatidyl inositol 3 kinase. Once that bind here, right, this will convert PIP2 into PIP3, right. And that's one thing. When this convert in this way in the cell membrane, PIP3 will increase. I want to mention some special thing, that there is another enzyme which can reverse this mechanism. That this can break down this phosphate and PIP3 can reverse into PIP2, and this is called P10. This is a phosphatase. Phosphatase means removing. Kinase is adding the phosphate. Phosphatase is removing the phosphate. So P10 is phosphatase. This is actually a molecular break on whole this pathway, P10. Now, anyway, move forward. PIP2 is converted into PIP3, and once cell has more PIP3, what will bind there? Naughty boy and the monkey. PDK1 act on AKT, and AKT will be phosphorylated. Now AKT will jump and do multiple functions. One function is what AKT will do. It will act on the intracellular vesicle loaded with GLUT4 and insert them to the membrane, and cell membranes become more permeable to glucose. Along with that, it will act on other components of the membrane also, increase uptake of amino acid, potassium and phosphate. Then, especially in the liver, enhances the glucokinase activity, and whatever glucose is coming in, it is going into phosphorylation. Remember, GLUT4 is inserted into muscles and adipose tissue. In the liver hepatocyte membrane, there are no GLUT4, they are GLUT2, which are not insulin dependent or sensitive. Remember, if we have liver here, muscle here and adipose tissue here, we add insulin to all of them. Muscle membranes become more permeable to glucose, adipocytes membrane become more permeable to glucose. Liver membranes are always permeable to glucose. It means permeability to glucose to muscles and adipocyte is insulin dependent because they are having special type of Glut4 inside the cells which will float on the membrane. But liver cells, they are having Glut2, right? Liver has a double function that when body is rich in glucose, liver should take up this glucose, right? And when body is poor in glucose, liver should release glucose. So liver cells should have two-way, what? All the time present Glut2. So Glut2 are not insulin dependent, but still when insulin is levels are high, liver is taking more glucose. Liver uptake of glucose is more, but not because liver cells got Glut4, no. Liver take up more glucose because when insulin bind with the receptors on the hepatocyte internal glucokinase activity increases and ability of hepatocyte to convert glucose into glucose 6-phosphate increases too much, and then when internal glucose levels are becoming less due to phosphorylation, external glucose will jump in. Again I will repeat. Tell me a tissue on which there are insulin receptors. There are insulin receptors, but glucose uptake is not insulin dependent. Hepatocytes. Which glucose transporters are there? Glut2. What are the tissues on which Glut4 are represented for glucose uptake after insulin exposure? Muscles, striated muscles and adipose tissue. Is that clear? Now, come back. So what I was talking about, that this Akt could bring Glut4 in muscle membrane and adipocyte, number one. Number two, Akt could in liver especially activate glucokinase activity, right? And in liver and muscle, it specially can phosphorylate GSK3 inhibited, so it is unable to phosphorylate GS, and GS unphosphorylated is more active and glycogen synthesis will start. Is that clear? With that, it has one more function, that it can activate, what is this? mTOR pathway, and this mTOR pathway will increase protein synthesis and also to some extent lipid synthesis. And when so much nutrition is coming and cells is taking up, what is this? More and more glucose and amino acids, and the synthetic functions machinery is doing well, right? More glycogen is coming, more proteins are coming, more lipid is being synthesized. Do you think cell is happy or sad? Happy cell. Happy cell should plan to live for long time or short time. Now, you know, our cells can decide either to live for long time, or our cells can decide they should commit suicide or not. You know that, or you don't know this? How you know that? Cells, every cell has inherent machinery to commit suicide. Within our cells, there are special proteins and enzymes. If they are activated, they will destroy our own DNA. And if this is a cell, right, almost every cell has special molecular machinery. If that is activated, cell will commit suicide. We say there is a programmed cell death. How? That those proteins, when they are activated, they lead to digestion and breakdown of the cellular DNA, and also digestion and disintegration and destruction of nuclear and cytoplasmic proteins. This process is called apoptosis. Is that right? When cells are damaged, right, and when cells are useless, they commit suicide. They undergo process of apoptosis. But when cells are happy, they want to live long. Now here I want to tell you one thing, that there are two types of genes which determine cell survival. There are two types of genes which are determining cell survival. Let's suppose this is a gene, right? This is a set of genes. There are many genes like this. The set of genes, when these genes produce their product, cell commits suicide, right? So we call this death process is called apoptosis. So these genes, when they are activated, they produce such enzymes and activate such enzymes which digest away the cell. So we say these are pro-suicide gene. These are pro-suicide gene, or we call it pro-apoptotic genes. In the same way in the cell, there are other genes. If these genes are activated, cell produce such product that apoptosis is inhibited and cell survival become long. Is that right? Now come back. When more nutrition, more insulin, right, intracellular machinery is working, right, and Akt is doing lot of functions, AKT is doing lot of function. One of the function is that cell survival should be decreased or increased? Cell survival should increase because cell is getting lot of nutrition. So actually AKT also act on special pro-apoptotic gene products. Some of the protein which are produced from here, they are really bad and they are also called bad, right? Bad genes produce bad proteins. Bad protein order the cell to commit suicide. And what happens? Akt will lead to phosphorylation of bad proteins, and when bad proteins are phosphorylated, they are sequestered and they are inhibited, and cell will not plan to commit suicide, right? So what did I discuss? I only discuss about that once insulin stimulates the receptors on the target cell, how phosphatidyl inositol 3 kinase protein, what is this protein? Kinase B or Akt pathway is activated. Now you have to repeat it with me, right? And after the break, we'll talk about other pathway, RAS, MAP kinase pathway. Listen, I told you when nutrients are more, insulin will act on the cells, target cells. Cell should have changes in 3 levels. Number 1, membranes changed. Then cytosolic enzyme metabolic machinery changed. And third is what? Genetic machinery changed. And all these actions are for anabolism, right? So up to now, we have discussed Pi3k Akt pathway or Pi3k P.K.B pathway. This pathway alters the membrane and alters the metabolism. And when we'll talk about RAS MAP kinase after the break, that pathway is mainly concerned with genetic expression of the alteration in genetic expression of the cells. This pathway, RAS pathway, which we'll discuss later, it is mainly concerned with determination of this happy cell should proliferate, differentiate, right, over survive, grow, right? This we'll talk later, but let's review it fast. Let's suppose I'm here, right? What is this coming now? First of all, lot of nutrients in the body, lot of glucose coming in the body, and pancreas, beta cells released insulin. Insulin bind with what? Alpha subunit. When alpha subunits are activated, they will activate beta subunit. Beta subunits will lead to autophosphorylation on what? Tyrosine residues. Is that right? Then phosphorylated beta subunit, they will attract IRS. Say loudly, they will attract IRS. Then this beta subunit enzyme activity will attack the IRS, and IRS will get phosphorylated. What kind of phosphorylation? Tyrosine kinase activity. Once IRS is, what is this? Phosphorylated, it will bring to special domain out, and it will allow other proteins to attach or allow the other proteins to dock in. That is why IRS is also called docking protein. What is it called? Docking protein. Other proteins can dock in. Now IRS, once it is activated, right, it will bind with a special enzyme, Tarzan 1. What is that? We'll talk about Tarzan 2 in the later part. What is it? Pi3k will convert PIP2 into PIP3. PIP3, what will bind it? Naughty boy, what is that? PTK1, and a monkey will come, AKT, and then monkey phosphorylated and activated. Now I'm the monkey. No, I'm really not the monkey, but just imagine that I'm the AKT protein or phosphorylated protein kinase B. Now I'm going to do monkey action. Number 1, take the vesicles and fuse it with the membrane, and membrane will become rich in what kind of transporters? Glut4, and glucose will come in. In the same way, I will also alter the permeability of the membrane for amino acid uptake, phosphate uptakes, and potassium uptake. So I will do membrane actions. this insulin and its receptor and molecular pathway changes in the membrane characteristics. Timeline is within few seconds to minutes. So membrane characteristics change within few seconds to minutes. After that, AKT or AKT, okay, this naughty monkey, AKT will act on in the liver, especially on which enzyme? Glucokinase, and enhance their activity or increase their expression. And that will lead to phosphorylation of protein intracellularly more, and more glucose will come in. Is that right? Thirdly, this AKT act on glycogen synthase kinase 3 and phosphorylated, and glycogen GSK3 will get inhibited, and once it is inhibited, what will happen? Then GSK3 cannot phosphorylate GS, and when GS is not phosphorylated, it is more active, and then GS will start synthesis of glycogen. So we have seen more glucose coming in, more glucose undergoing phosphorylation, and more glucose going into glycogen synthesis pathway. Because anabolic activity is occurring, so anabolism and catabolism do not go together simultaneously. So if glycogen synthesis is going up, what should go down? Glycogen breakdown should go down, and also gluconeogenesis should go down. Then ACT, number 1, it changed the membrane. Number 2, it was changing glucose handling. Now, and carbohydrate. Now this ACT will act on another protein called mTOR, and mTOR will alter and lead to metabolism of protein and lipid. And under these nutrient rich situation, what should happen to proteins? Synthesis should go up, and lipid synthesis should go up. So membrane characteristics altered and metabolism also altered. Membrane characteristics were altered within seconds to minutes, but metabolic activity or enzyme activity is altered over minutes to hours. So these activities within seconds to minutes, and other activities within minutes to hours. Is that right? And then even action is occurring on, what is this? On the pro-apoptotic machinery is inhibited. Pro-apoptotic machinery is inhibited. So cell can take more glucose, can phosphorylate more glucose, can make lot of glycogen, can make more proteins, can make more lipids, and then enjoy a long life by inhibiting pro-apoptotic business. Is that right? So whole this was over few minutes to hours. And then after the break we will discuss this pathway. IRS will activate another pathway on this side, which I will draw, and that is basically RAS MAP kinase pathway. That pathway will lead to cellular growth, differentiation, proliferation, and survival. That pathway changes the genetic machinery of the cell, right? And that pathway takes hours to days. So membrane changes seconds to minutes, metabolic enzyme changes minutes to hours, and genetic expression changes hours to days. Let's have a break.